Hello, everyone, and welcome to our February Clinical Culture and Diversity series. Our presentation today is the complexity of triple-negative breast cancer in African American women. The Clinical Culture and Diversity series is a collaborative quarterly series by the Diversity, Equity, and Inclusion Cabinet, the Office of International Programs, and the School of Medicine's Office of Diversity and Inclusion. Our series explores issues related to intercultural communication in the clinical setting, developing cross-cultural competencies, improves patient care, provides individuals with a comprehensive educational experience, and helps KUMC become a more diverse and integrated campus. Students, faculty, and staff are invited and encouraged to contribute to the conversation. We do have one more series scheduled for 21-22, and that will be on Tuesday, April 19th. We do have continuing education credit for today. I have shared those slides in the chat, and I will also share that information again for those who joined after. If you do want continuing ed credit, you do need to make sure that you've signed in with your first and last name. Here's just some more CE information. Again, I'll share these in the chat, so don't worry if you don't get these down. And now our introduction of our speaker, Dr. Joan Lewis-Wamby by Pamela Scott.
Hi, everyone. I am Pamela Scott from the Office of Diversity and Inclusion, and I have the honor of introducing our special guest speaker for this third component of our CCDS workshop series. Dr. Joan Lewis-Wamby is an associate professor, tenured, and the director of graduate studies in the Department of Cancer Biology at KUMC. Dr. Lewis-Wamby received her PhD in cancer biology in May of 2002 from Rutgers University, and she completed her postdoctoral training at Northwestern University and at Fox Chase Cancer Center. Dr. Lewis-Wamby has worked in the breast cancer field for over 15 years with expertise in estrogen receptor signaling, endocrine therapy resistance, triple-negative breast cancer, and cancer health disparities. The focus of her lab is to understand the mechanisms by which breast cancer cells and breast tumors develop resistance to endocrine therapies and to develop alternative therapies for breast cancer patients. She utilizes unique human breast cancer cell lines, animal models, and clinical breast cancer samples to uncover the molecular underpinnings that drive the development of resistance in breast cancer patients. Additionally, her lab also studies triple-negative breast cancer and inflammatory breast cancer, which are two aggressive and lethal subtypes of breast cancer that are difficult to diagnose and treat, and disproportionately affect African American women compared to other ethnic groups. Dr. Lewis-Wamby has published over 45 research articles, and she has received several honors and awards, including NIH Young Investigator Award, Avon Scholar, Susan G. Komen Scholar, WIMS AAA or AAMC Scholar Award, and the KUMC Rising Star Scholar. Her research has been funded by grants from NIH, ACS, and the Department of Defense. Please help me in welcoming Dr. Joan Lewis-Wamby.
Okay, thank you, Pamela, for that wonderful introduction. You make me sound better than, than I
As you are.
So I'm going to share my screen, hopefully. Okay, let's see. One sec, I need to go back. Okay, can you guys see my screen? Great. I will minimize. Okay. So thank you, Pamela, for inviting me to present as part of your clinical culture diversity series. I think this is my first time presenting. I think I've participated in other panels, but this is my first time being a part of the series. So I'm happy to be here. Thank you all for spending this next hour with me as I share with you some of the work that we're doing in the lab. And I will start by just starting with the brief overview of breast cancer, and then I will get into specifically what we do in the lab. As some of you may know, breast cancer is the most diagnosed cancer among women in the US. And it is the second leading cause of cancer death. It's second to lung cancer. In 2022, it's estimated that over 287,000 women will be diagnosed with invasive breast cancer, and over 43,000 will die this year from the disease. We know that currently over 168,000 women are living with metastatic breast cancer, and worldwide we have over 3.8, close to 4 million women who are survivors. We've done tremendously well over the years in terms of improvement in treatment for breast cancer. The survival has improved over the years. And so the 5-year survival for early stage breast cancer is 90%, which is extremely good. But we also know that it is not, the 5-year survival is not as great for African-American women. Unfortunately, we know that 40% African-American women have 40% higher mortality rate compared to other groups, even though we have a lower incidence of breast cancer. So there's a lot more work that we need to do in that regard in closing that disparity gap. So one of the things that whenever I talk about breast cancer, people always ask, well, what are some of the risk factors, or how do we know who's at risk and who's not at risk? And what I always say is that breast cancer is a sporadic disease. So there's no one factor that causes it. There are multiple factors that may contribute to increasing your risk of getting the disease. And genetics is part of that. We know that the biggest risk factor is your gender. So women are more likely to develop breast cancer compared to men. It's a disease of aging. So the older you are, the higher your chances or your risk of getting breast cancer. If you have a family history of breast cancer, that will increase your risk. If there's a genetic mutation, so BRCA1, BRCA2 mutation, that will increase your risk. And we know for certain subtypes of breast cancer, there's an increased risk with race. So African-American women are more likely to develop triple negative breast cancer and inflammatory breast cancer compared to some other groups. So those are the unavoidable risk factors, things that we can't change. But we also know that there are lifestyle factors that can also contribute to increasing your risk. And those factors include obesity, high alcohol consumption, nulliparity, meaning you've never had any children, hormone replacement therapy. And the biggest thing is when we talk about gender and HRT and a lot of these other factors, it is your lifetime exposure to estrogen that really increases your risk of getting the disease. And so lifetime exposure comes from age of menarche, age of menopause, exposure to environmental estrogens, HRT and oral contraceptives, and also obesity, because obesity is one of the main drivers of estrogen production in postmenopausal women. And we know that as we age, we tend to gain more weight, and that can also increase our risk of getting the disease. So it's also important to remember that breast cancer is not a uniform disease. It's actually a disease of subtypes. And so we define breast cancer based on its expression of certain types of receptors. And so we know that breast cancers can express the estrogen receptor, and that represents about 60% of all of the breast cancers that are diagnosed. And so those are termed the luminal breast cancer because they express the estrogen receptor and the progesterone receptor. We have breast tumors that express HER2, which is another growth factor receptor, and that represents about 10 to 15% of the cases. And then we have the triple negative breast cancers. And so for triple negative, these are breast cancers that do not express the estrogen receptor, progesterone receptor, or HER2 receptor. So they lack these three main receptors that we usually use to diagnose patients and then determine treatment options for them. So understandably, the fact that these patients do not have ER, PR, and HER2, there are less treatment options available for patients who are diagnosed with triple negative. So, like I said, the majority of breast cancer is estrogen driven, and estrogen works through the estrogen receptor, which is usually expressed in about 70% of those tumors. And so these tumors rely on estrogen for growth. And if you take away estrogen, then these tumors can no longer grow. And so that is the majority of the breast cancers that is diagnosed among women. And so, as I mentioned before, the presence of these receptors are used to determine the types of treatments that patients receive. So if you're ER/PR positive, which is the majority of cases, those patients are treated with endocrine therapy, and I'll talk a little bit more about that. But those patients, their overall survival is very good. So you can see it here. This is a 5-year survival curve. The blue line represents the luminal breast cancer. So luminal A, it's ER positive, PR positive tumors. And so those patients fare the best. So they have an overall survival between 80 to 90%. That's followed by the luminal B subtype, which is ER positive, but sometimes those tumors lack the progesterone receptor. But luminal breast cancer, which is A and B combined, they represent about 70%. And so the overall survival ranges from 80 to 90% for that subtype. And that's followed by the basal subtype, and this is the triple negative, which I said the triple negative, they lack ER, PR, and HER2. And as expected, since there are no known targets for that subtype, the overall survival is much less compared to ER positive tumors. And for HER2 positive tumors, those patients tend to do well somewhat, but they also have the case of relapsing, and so overall survival is less for HER2 positive tumors as well. But overall, what this shows us is that if your tumor expresses ER, PR, and HER2, there are targeted therapies that are available for those patients, and overall their prognosis is very good. If it's triple negative, there are less targeted therapies available, and so those patients are treated with the standard of care, which is surgery, chemotherapy, and possibly radiation. And overall, the survival rate for triple negative is less than for those that are diagnosed with ER positive breast cancer. And so this slide just shows you the different therapies that are available if your tumor is ER positive versus if it's HER2 positive. So if it's ER positive, we have endocrine therapy or hormonal therapies. And these drugs, they either target the estrogen receptor, which is what is needed for estrogen to activate. And so these are called estrogen antagonists or anti-estrogens. And so they bind to the estrogen receptor and they, in effect, inactivate the receptor so the receptor can no longer stimulate the growth of these cancer cells. And then we have estrogen suppressors. These are aromatase inhibitors here. And currently we have 3 that are FDA approved for the treatment of breast cancer, anastrozole, exemestane, and letrozole. And these are estrogen suppressors. And what they do is they completely suppress estrogen production in the body. And they do that by binding to the aromatase enzyme, which is the main source of estrogen production in a woman's body once you reach menopause. And so aromatase inhibitors will block the aromatase enzyme and prevent it from synthesizing androgens to estrogens. And so in a sense, you're creating an estrogen depleted environment where the cells no longer have access to estrogen in order to grow. So that is the main strategy that's used to treat ER-positive breast cancer, and those strategies have worked really well. Like I said, the 5-year survival for ER-positive early stage breast cancer, it's about 90%. Patients who are diagnosed with late stage disease or metastatic disease, their overall survival is much lower. So it's important, obviously, to detect the cancer early enough so you can start treatment. And so that will increase your overall survival. For HER2 positive tumors, currently the main treatment is trastuzumab, which is a monoclonal antibody that targets the HER2 receptor. So these main strategies have worked effectively for the ER positive tumors and HER2 positive tumors. But my lab is interested in not only studying ER positive tumors, but also triple negative. And for the purpose of our talk today, I'm going to focus more on triple negative because it's really an area that we're understanding more about triple negative, but we're way behind in terms of developing targeted therapies for triple negative. So it is a complex disease compared to ER positive. It's a highly heterogeneous disease, which means that there are many factors that contribute to its aggressive phenotype. Whereas ER positive tumors are driven by the estrogen receptor, so it makes it easier to target the estrogen receptor for therapeutic purposes. For triple negative, there's no one factor, there's no one pathway that we've identified that it's driving the aggressive phenotype of triple negative. So there's a lot more work to be done, and there are lots of labs that are studying triple negative, some in the context of health disparities. Some labs are just looking at triple negative in general. But for my talk, I will focus on triple negative in regards to African-American patients. So we know that triple negative has very few treatment options, and there are no current targeted therapies that are available. So the standard of care for triple negative, it's surgery, chemotherapy, and possibly radiation, depending on the stage that you're diagnosed with the disease. We know that triple negative also disproportionately affects African-American women compared to other ethnic groups. It also affects younger women. So it's actually a disease that we see in younger women compared to ER positive. ER positive disease tends to be diagnosed in women who are older. So 55 and older, triple negative, it's usually in your 30s and in your 40s. So it's a disease that affects younger women. And it usually occurs in women who also have the BRCA1 gene mutation. And so BRCA1 mutation will increase your risk of getting breast cancer as well as ovarian cancer. So we've seen that for triple negative, there's a higher percentage of women who have the BRCA1 mutation as well. As I mentioned, African-American women are 3 times more likely to be diagnosed with triple negative compared to Caucasian and Hispanic women. And we also know that they have a higher mortality rate compared to other groups, and a higher mortality rate, not only for triple negative, but also for ER positive breast cancer. So this is some of the clinical data that shows that disparity. And you can see from this graph here, they were comparing African Americans to European Americans in terms of the percentage of triple negative. And you can see that it's clearly higher in African American patients, and it's very high in younger women. So younger women, African American, have a higher risk of being diagnosed with triple negative breast cancer compared to their European counterpart. And you can see the overall survival for ER positive versus triple negative. So ER positive, the overall survival is much better compared to triple negative. But you can see a big difference between overall survival for African Americans compared to their European counterpart. So there's a big difference in overall survival between the two groups. But we also know that a higher incidence of triple negative is not just unique to African Americans here. They've done some clinical studies where they've looked at triple negative in women of African descent. So you have women in East Africa, North Africa, and West Africa, and you can see higher levels of triple negative in these women of African descent, which is even higher than what you see in African Americans. So it's definitely a disease that affects women of African descent. And in the United States, we know that that higher incidence, it's related to many factors. Obviously, in other countries, there may be other contributing factors to the higher incidence and disparities that you see there. But in the United States, we know that we try to focus on what is driving this disparity that we see in terms of overall survival, but also in the higher incidence of the disease. So one of the major questions that we always try to address is triple negative higher in African-American women because of the biology of the disease? So you have a higher incidence of the disease, and is it more aggressive just because of the biology of it? And is that what's contributing to the high mortality rate? Or are they other factors, social factors, that are impacting the overall mortality rate in those patients? And so our lab is really focused. We don't look at the social factors. We look more at the biology of the tumor and try to understand what is happening at the molecular level. But there are a lot of studies that are focused on socioeconomic factors that may contribute. So lack of access to care, lack of access to clinical trials, poor screening, just factors that has nothing to do with the biology of the tumor, but can impact overall the types of treatments the patients are able to receive, how well that they're able to get access to maybe novel treatments or clinical trials that may be more relevant to their disease. So those are factors that, those are things that we can look at and modify in some sense. And biology is something that, it's not modifiable. It is what it is, but sometimes it's very hard to tease out biology versus environment. So I think it kind of works together to kind of generate this disparity that we're seeing in terms of overall survival that we witness for triple negative. So our lab is really interested in understanding the genetic factors, the biology of the tumor, and how does that biology alter triple negative to make it more aggressive, but not neglecting the fact that environment also impacts overall survival and how well patients respond to treatment, the types of treatment that they have access to as well. So really, we're interested in addressing this unmet need in triple negative, really trying to understand the biology of the tumor, identify the factors driving its aggressiveness, and then to see whether or not we can identify an effective target so that we can impact the treatment options for patients. So that is the goal of our lab. And so we want to study triple negative as a disease, as the totality of the disease, to understand the drivers of its aggressive phenotype. We also want to identify therapeutic targets that we can use to potentially treat triple negative. And right now we're focusing on triple negative in African-American women, just because we've identified this unique feature that we think that we can exploit and make progress in that area. But overall, as a lab, we're interested in triple negative as a disease because it is understudied and there's a lot we still don't know. The focus of our lab is really to have this bench to bedside development in terms of our research pipeline. And so our idea is to develop critical models that we can use in the lab to investigate and try to figure out why these genetic differences may occur, and then how can we then translate that finding into the clinic? How can we use that information ultimately to benefit patients? And so we do basic research, but there's a big translational component to our work, because ultimately our goal is to translate our research findings into clinical applicability. And so one of the things that we did early on as we started out was to really try to obtain clinical samples from KU Med. So we have a biosample repository here at the Med Center, and they collect tumor samples from patients who are diagnosed with breast cancer. And so when I initially joined, I was interested in ER positive tumors as well as triple negative. And I had the opportunity to collect triple negative samples from African American patients, from Caucasian patients, and normal control. And we did some screening studies, molecular studies, where we were characterizing genetic differences between these genes. And one of the genes that we identified that was uniquely expressed or differentially expressed in African American patients compared to Caucasian patients was this gene called IFIDM1, IFITM1, which is an interferon stimulated gene. And what we found was that this gene is highly expressed in African American triple negative tumor samples, but not expressed as highly in triple negative from Caucasian patients, and it's not expressed at all in normal tissue samples. And so since we're interested in identifying unique pathways that might be driving this aggressive phenotype for African American patients, we're always interested in what's different in these tumors, what's different compared to other tumor types. And so since we saw that IFITM was overexpressed, we then did some overall survival data to see whether or not overexpression of IFITM1, if it correlates with survival. And what we found was that patients who had high levels of IFITM1 had a lower overall survival rate compared to patients who had low levels of that. And this is for triple negative breast cancer. We also had the fortune of collaborating with an investigator at the University of Tennessee, and he does a lot of health disparities work. And he was able to provide us breast cancer cell lines that were generated from African American patients, and also triple negative cell lines that were generated from Caucasian patients. And we then looked for this gene and protein that we identified in our clinical samples that seemed to be uniquely expressed in African American tumor samples. And what we found was that in the triple negative breast cancer cell lines from African American patients, and these are the 3 cell lines from African American patients, our protein of interest is highly expressed in those triple negative cell lines. And if you compare it to the triple negative cell lines from the Caucasian patients, it's not expressed. So this data suggests to us that it might be a unique marker for triple negative, but especially in African American patients. We also took these cell lines and we measured interferon levels in these cell lines. So interferon is one of the major drivers for IFIDM1, and I will cover that in a few slides later. But we also measured interferon alpha levels in these samples, and we found that it was significantly higher in the triple negative cell lines from African American patients, but not in the triple negative cell lines from Caucasian patients. So this suggests that the interferon signaling pathway and IFIDM1 might be a unique marker for triple negative in African American patients. Obviously, there is a lot more for us to do to validate that as a potential target. We were also able to collect clinical samples from our collaborator at the University of Tennessee. And this is from patient-derived xenografts. And so patient-derived xenografts, these are samples that were taken from patients and then put directly into animals. And then we look and see whether or not these patient-derived xenografts, if they're able to develop tumors over time. And so from this patient sample, we were able to see that IFIDM1 is highly expressed in the PDX from African American patients, but not expressed for Caucasian patients. And we looked at some other markers as well, and those markers are also elevated in African American patients. So what is the function of IFIDM1 and what is it doing in our triple negative cell was the next question that we wanted to address. So what do we know about IFIDM1? So IFIDM1 belongs to the family of IFIDM proteins. And so these proteins are mainly known in virology. So they've been well studied in influenza and HIV and some other viruses. And what these proteins do, they block viruses from entering cells. And their goal is really to help the cells survive. So we don't know what it's doing in cancer. And specifically, we don't know what it's doing in our triple negative cells. And so we're trying to understand the function of IFIDM1 and its mechanism of action and whether or not we can potentially target it in our triple negative cells. So that is the goal of what we're doing in the lab. And so as I mentioned before, interferon alpha, so interferon is a cytokine. It's usually activated upon viral infections. And so it stimulates the immune system. It helps us to fight off viruses. And so normally in normal cells, it's not activated unless there's some virus that it's fighting off. But in our cancer cells, and especially in our triple negative cells, IFIDM1 seems to be just constitutively active. And so this pathway seems to be activated all the time in our cancer cells. And so what we're trying to understand is why is it activated? And can we potentially block this pathway to see some type of therapeutic benefit? So IFIDM1 activates JAK-STAT signaling, and JAK-STAT then activates a whole host of interferon-stimulated genes. And these genes are important for cell survival and for activation of the immune system. So one of the questions that we're trying to address is why is this pathway activated in our triple negative cells? But specifically, why is it activated in triple negative in African-American patients compared to our Caucasian patients? And specifically, why is IFIDM1 elevated and not other markers of the interferon alpha signaling pathway? We know that when we block interferon in these cells, so if we use a neutralizing antibody and prevent interferon from interacting with its receptor, we can significantly decrease IFIDM1 expression in these cells. And so this gives us additional proof that IFIDM1's expression is really driven through activation of the interferon signaling pathway in these cells. So what do you do? So you see that this gene is elevated and you want to know its function. What we normally do is we usually knock the gene out if we want to know its function. And so we use this technique or genetic editing technique called CRISPR. So we CRISPR'd out the IFIDM1 gene in our triple negative cell line. We knocked it out. And then we looked at the effects of this knockdown on the ability of these cells to grow and their ability to form tumors in culture. And so these are markers of tumorigenesis, the ability of the cancer cells to grow, the ability of these cancer cells to form anchorage dependent growth, and their ability to migrate in culture. So these are all hallmarks of cancer. And so we utilize different assays to look at the potential of these cancer cells to not only proliferate, but to also migrate and invade, which are critical steps that you need in the progression of cancer. And so you can see that when you knock down IFIDM1, these cells, it significantly reduces the ability of these cells to grow. It reduces the ability of these cells to form colonies, and it reduces the ability of these cells to migrate. So normally how we look at migration, we take these cells, they're grown on special flask or plates that allow them, so these are cancer cells that adhere to the plate. We then make a mark on the plate with a sterile pipette tip, and then we look at the ability of these cells to migrate towards the mark or the line that you draw, and then we measure how quickly those cells are able to then migrate to that mark. And that tells us how quickly the cells are migrating. So it's just a rough way of estimating the migratory and invasive potential of these cancer cells in culture. So we also took those cells, we injected them into animals. One of the things that we do in the lab as well is we do a lot of animal studies. So we do cell culture studies where we take breast cancer cells, we grow them in culture, and so we can keep these cancer cells growing for a long period of time. But we can also take these cells, we can put them into animals and see how well the cancer cells are able to form tumors in vivo. So it's always important when we have an important observation in culture to make sure that we're demonstrating that the same thing can happen in animals. So to demonstrate the in vivo potential of our in vitro findings. And so we took these cells that we knocked down IFIDM1, we injected them into mice, into the mammary fat pad of the mouse, and then look at how well these triple negative cells are able to form tumors in these mice. And you can see here that if you knock down IFIDM1, the tumors are a lot smaller. And so they have their ability to grow in the animal is significantly reduced compared to the control cells. And so the control cells are expressing high levels of IFITM1, and the knockout cells do not express IFITM1. And so the loss of IFITM1 is inhibiting the ability of these animals to really form tumors, triple negative tumors. And we can see here, we did another assay to kind of verify, well, why aren't the tumors growing? So tumors can grow if you inhibit their ability to die. And so you can see here that if you knock at IFIDM1, there's an increase in cell death through tunnel staining. And this just stains cleaved DNA in the cell. And it's a marker of cell death. So if the cells are dying, that's why the tumor is not growing. So loss of IFITM1 suggests that these cells need IFIDM1 in order to stimulate their growth. And if you knock out IFIDM1, then the cells are no longer able to survive, and so they start to die. And this was another assay that we used to look at the invasive potential. So I mentioned before the ability of the cells to grow in tissue culture. We can look at growth, we can look at migration, we can look at invasion, but we can also do the same thing in vivo, so in animals. And so this model that we're using, the MIND model, we can look at how well the cancer cell is able to invade out of the mammary duct into the surrounding tissue, and that tells us how invasive the cells are in vivo. And so this is a good marker of how aggressive the cells are. So if the cells are not able to grow, if they're not able to migrate, if they're not able to invade, that's good because that makes them less aggressive and easier to treat. If the cells are highly proliferative, if they're able to migrate and invade, that means that they have a high potential to eventually metastasize. And most breast cancer patients die from metastatic disease. They don't die from primary cancer. They die from the cancer migrating from the primary site, which is the breast, and going to different organs. So the lung, or going to the bone, or going to the liver. So metastatic breast cancer, that's what most patients die of. So this data shows us that IFITM1 overexpression is important for the survival, growth, invasion of these cells. And if it's not there, you can significantly reduce the aggressive phenotype of these cells, not just in culture, but also in animals, which is very encouraging. When you're doing research, you like to see that your in vitro findings are replicated in vivo in animals. And so another part of the research that we're doing is, now that we've identified IFIDM1, we're interested in determining if there is a way for us to specifically target IFIDM1 in our cancer cells. And so we do a lot of high throughput screening. We have access to thousands of FDA approved compounds, and we collaborate with KU Lawrence and Dr. Scott Weir, who's in drug development and therapeutics. And the goal is really to screen drugs that have already been approved for FDA, and maybe not approved for breast cancer, but some other disease, and whether or not we can repurpose that drug or those drugs to target IFIDM1 in our triple negative cells. And so we've started this project and we've identified one compound, parthenolide, which is an NF-kappa B inhibitor. And it can inhibit IFIDM1, and we're starting some studies with it. But ultimately, the goal is to identify drugs that could potentially target IFIDM1 that have been FDA approved, but they also have low toxicity profile as well. And that would increase their chances of getting into the clinic for a clinical trial. And ultimately, that is the goal, to see if we can translate our findings in the lab back to the clinic. So we've started some animal studies with this drug that we identified, parthenolide, and it does seem to work in our triple negative model that we've developed with one of the triple negative breast cancer cell line from our African-American patients. So that's very encouraging for us. Going forward, we want to combine parthenolide with some other drugs that are currently available. Ruxolitinib is a JAK1 inhibitor. They've tested it in ER-positive breast cancer, and there are some clinical trials testing ruxolitinib on triple negative in a subset of breast cancer patients. So the idea is maybe we can combine parthenolide with ruxolitinib and some other drugs to see if it would be therapeutically beneficial to these patients. But ultimately, our goal is really to target IFIDM1, and if we can develop a small molecule that can target IFIDM1, that would be ideal. But for now, the goal is to really use compounds that have already been FDA approved, and to see if we can do combination treatments with these different drugs. So really, the idea is we have this data that shows that IFIDM1 is overexpressed in our triple negative cells, that it's overexpressed specifically in triple negative from African-American patients, and that its overexpression is associated with the aggressive phenotype of triple negative in that patient population. We also show that targeting it has therapeutic benefits as well. Our goal really is to try to see if we can utilize IFIDM1 or other factors to develop as specific biomarkers for triple negative. We will start with the African-American patients, but ultimately our goal is to try to see if we can identify biomarkers for triple negative in general. IFIDM1 seems to be a good marker for African-American patients, but I think we will need access to larger patient samples. So right now I'm collaborating with investigators at the University of Tennessee, and they have access to a larger cohort of African-American triple negative samples. And so the idea is to really, because in-house we have access to maybe 50 triple negative samples, but the idea is to look at a larger patient samples, possibly 200 triple negative, and then match that with Caucasian patients and normal tissue samples to see if it's truly a biomarker, and then to see if we can also get clinical samples. So apart from tumors, get serum from those patients to identify other markers that might be present in the serum for those patients. And so for serum, we have access to serum from patients here at the biosample repository. We also have access to patient samples at Truman Medical Center and then University of Tennessee. So going forward, that is the goal, to get access to a larger set of patient samples and to see how we can use that information to really try to identify not only drivers of triple negative, but if we can potentially identify a therapeutic target for the disease. And another part of what we're doing is trying to share our laboratory findings with others. So the Cancer Center is part of PIVOT, and PIVOT stands for Patient and Investigative Voices Organizing Together. And what that is a program that gets survivors together along with basic scientists, and we work together and we kind of bring them on board so that they can see what we're doing in the lab and they can bring the patient perspective as to why we should be doing what we're doing, or if we're not doing the right thing, kind of say, you know, maybe you need to focus on this, or maybe focus less on that. I think it's always important for us to have that voice to help us to realize why we're doing what we're doing and whether or not what we're doing, the type of impact it would have on actual patients. And so we work really close with the members of Pivot, which has been great. And they really help us in terms of putting together projects. So a lot of times we have ideas for projects that we want to submit for grant funding. And so Pivot will be a part of that process. They will meet with us, we will present to them our ideas, and they will give us feedback. And they just help us to make sure that we always keep our grants and our research patient-centered. And I think that's always important for us as basic scientists. Since we're not directly interacting with patients as clinicians are, I think it's important for us to remember that, you know, what we're doing, it has implications for patients, and to have their feedback really helps us in focusing on what we need to focus on and prioritizing our research topics and just kind of honing in on the questions and strategies that we're doing and how that could be beneficial to others. Pivot also helps us in terms of just disseminating information to the community. And so the members of Pivot, they come from our community, they are from Wyandotte County and other areas. And I think they bring this unique perspective to what we're doing and what's important to not just them and their families, but their friends and other people in the community. So they can help us disseminate the information and bring more awareness to the types of research that we're doing in the lab. And so that's a big part of what we do in the lab, but also part of the Cancer Center. So I think it's very important that we should have public awareness of our research and sharing that information with others, with our lay audience, so that they can have a better understanding of what goes on in the lab and what we're researching. We have a lot of our Pivot members, they come and they spend time in the lab and they see hands-on the types of experiments we're doing. They're part of our journal club meetings and our lab meetings, and they do have opportunities to really interact with us and interact with our graduate students and our faculty and other staff. And so it's great for them, but it's also great for us as well. So I'd like to take this chance just to acknowledge all the people in the lab who have done the work. And so a lot of the work was done by my graduate students, Asona, Olivia, and Taylor. And so they just finished. Olivia, so Taylor is a recent postdoc. She just started a postdoc at Northwestern. Olivia is doing her postdoc at Yale. And Asona is an MD PhD student. And so she finished and now she's doing her residency at the University of San Diego. So, you know, I think for us, we have the opportunity to not only do research, but we have the opportunity to disseminate our findings with others. But it really takes a team. And so, you know, our collaborators really help us. Obviously, my students help with all these projects. The core facilities here, the biosample repository that provides us tumor samples, the animal facility that allows us to do our animal experiments, and also the members of Pivot. They've been really helpful in helping me and helping so many others in the Cancer Center really focus, keep our research patient-centered, and also our funding agencies. So I thank you, and I will take any questions. Do I stop sharing or?
It doesn't matter, Dr. Wamba, you can leave it up or take it down. And Brianna's gonna fill the chat to see if anyone has any questions that we can direct towards you to answer.
And feel free to unmute yourselves if you have questions you want to ask that way as well.
So I have a question for you, Dr. Wambi. What got you initially interested in pursuing breast cancer research?
Um, I think I've always been interested in science. um, in terms of breast cancer, I think growing up, I really didn't have a clue as to what breast cancer is. And I know we had some family friends and family members who were diagnosed with breast cancer. And I think not knowing a whole lot about it, it kind of drives a lot of fear, because you don't really know a whole lot about the disease. And for me, I think it was really empowering for me to learn more about breast cancer. And since I had several like close friends or family friends who were diagnosed, and it just felt like I wanted to know more about it so I can help them. And so not just help myself, but to help them to not be so afraid. And I think the more you know about something, I think the less fearful you are, because you're kind of, you feel equipped with knowledge, and that helps you. And so since high school, once I got to college, I kind of was looking for opportunities to do research. And by coincidence, I ended up in a breast cancer lab as an undergrad, doing my senior research thesis. And I really loved the lab. It was such a great experience. And I was there working with other graduate students. And I wasn't a graduate student, I was an undergrad. But the graduate students were so engaged, and they really helped me, and they provided that positive environment to make me see like, oh, this is what grad school is about. It's not, because you have this perception of what getting your PhD is really like. But then once you're in a lab, and I'm still an undergrad, I was like, I had no idea. But it was just a positive experience to just learn from them and to learn more about the disease. And once I learned more about it, I felt like, okay, at least I know more about it. And if someone in my family asks me, I can help them better understand. And so the fear is always there. Whenever you hear cancer, it's going to be, it drives fear. But I think it's less fearful. It gives you more hope once you know the types of treatments that are there, the types of clinical trials that are available, and you meet other survivors. I think it just kind of, once you're exposed to it, you realize that it's not so scary. I think not knowing is probably the biggest driver of just fear and inaction. Not wanting to go get your mammogram because you're afraid, what if they find something, or not wanting to go get treatment because you don't know, it's just, fear is a barrier to so many things. And so early on, I just wanted to be in a position where I knew more about the disease so I can really help those who were going through it and really didn't know a whole lot about it. So.
Okay.
But I know it was like highly like basic sciencey, and because that's what I do. It's kind of hard to make it more. I was trying to see how I could make it less basic science, but that's all we do in the lab. We like knock out genes and, profile cells and do like all this stuff. So, but like overall, the idea is really not just to do the experiments in the lab, but ultimately to see how can we then disseminate that information. Because I think one of the things going through Pivot, I met so many patients who didn't know like about research, what we were doing in the lab. And it really helped them to have a better perspective of research, and even having them come to the lab and seeing like what we do has been really eye-opening. So yeah, it's good to do the research, but I think it's also super important to make sure that you're engaging in the community and you're bringing others along so that they understand what you're doing and why you're doing it. So.
Dr. Lewis-Wambi, thank you so much. This is Geraldine for a great presentation.
Oh, hi, Geraldine.
Good to see you. Yes, it is basic science, but many of us on here love basic science. So I know I was intrigued by your presentation. I do have a question about how do you see this translated into practice more? And not that it's not being, but I'm just wondering your thoughts about that and how could we have a greater impact on the women who are, as you stated, disproportionately affected by cancer?
Oh, absolutely. I think one of the challenges we face, like our goal is really to identify a biomarker that we can use to say, hey, if patients come in and their tumors express this biomarker, this will tell you how well they'll respond to treatment or not respond to treatment. So ultimately, we want to be able to translate what we're finding in the lab into the clinic. In terms of how that impacts treatment, I'm not sure at this point. We know that for targeting the JAK-STAT signaling pathway, we have a drug that's available right now that's FDA approved for something else, but currently they're looking at ruxolitinib, which is a JAK inhibitor. They're looking at it in triple negative. I think the challenge with a lot of these clinical trials is that we do not have enough African-American patients involved in these trials. So a lot of these trials, they're great and we need them, but they're not representative of the patient population that we're trying to reach. And so one of the things that we have to do is to get more African-American patients involved in clinical trials. And that's hard. That's hard to do if they're not aware of those clinical trials that are going on, if they're not comfortable participating in those clinical trials. And so that makes it even harder for us to really identify a marker because we don't have the tools to do it. We don't have the patient samples to do it. It was so hard for me to do these experiments because I had to go outside of KU because we don't have that many triple negative from African-American patients. And so it's just like, I think it's lack of access to those samples, lack of access of patients who are going to be able to participate in those trials that really do need to participate. But I think we are encouraged. I think if we can, right now, I'm collaborating with another scientist in the cancer biology department to see if we can have nanotechnology to detect certain biomarkers in the serum and to detect it at a really low level. And will that be easier to do, using this nanotechnology? And we're proposing to write a grant for that. But I think it's multi-layered and it's not one, there's no one answer to fit that. I think we do have to do the work in the lab to identify these potential markers, but we do need to have the clinical trials also to test it, to make sure that these are viable markers. But we need to have access to those patients. We need to have access to those samples to be able to do it. And, some places have more access than others, but we're making progress. So
Thank you.
There's a question in the chat from Danielle. Are you collaborating with any other researchers across our geography? One researcher who came to mind is Dr. Green in St. Louis.
Oh no, I don't know who's Dr. Green. No, I know a couple of clinicians at WashU, but and we're collaborating on endocrine resistant breast cancer, not triple negative. So I think not knowing who's doing what, I think that's also a challenge, and getting others to actually want to collaborate, because a lot of times people say they want to collaborate, but they don't follow through on it. So, and I've had great luck pairing up with other basic scientists or clinicians who are really interested in collaborating. So we're working from there. But yeah, I mean, I'm always open to collaborating with others because I think when you widen your circle, you get more ideas, and that's how you grow science. So I'm always interested in collaborating. So
I see a lot of comments, but I think that's it for the questions. Dr. Wambi, I just want to thank you for presenting today. I found your presentation to be very informative, and I think everyone else did as well. And thanks, everybody. Have the good rest of the day, and we'll see you again in April for the last CCDS.
Well, thank you, Pamela, and thank you, everyone, and thank you for the opportunity to present my work. So,
yes,
all right,
thank you.
guys.